Hey guys, and welcome back to Biologue. Hope you're having a great day and a great week. In this video, we're going to be looking at a walkthrough for the May-June IGCSE 2023 Paper 6 paper for Biology. So if you guys like this video, make sure to like, share, and subscribe to Biologue. And without further ado, let's begin. Alright, let's get right into it. So some animals live in cold places. They tend to huddle together as shown in the figure 1.1. A student investigated the effect of huddling on heat loss from modern animals. Test tubes containing hot water represented the animals. The student used the method described in step 1 to step 10. Step 1 was place a test tube in an empty beaker and put a thermometer into the test tube. Step 2, approximately half fill the test tube with hot water and start the stop clock. Step 3, after 1 minute record the temperature of the hot water in the test tube. Okay, so first question most likely will be, record the temperature as shown in the figure. So as you can see, this is approximately your 65, 66, 67, 68, 68.5 approximately, I would say. So that is your answer, 68.5. And degrees Celsius is already put for you. Record the temperature of the hot water in the test tube every minute for a total of 5 minutes. Put the 3 identical test tubes together and place them with an elastic band shown in the figure. Place the group of 3 test tubes in an empty beaker. Half fill all the 3 test tubes with hot water and put a thermometer into one of the test tubes. Record the temperature in the test tube at 1 minute intervals. Put 7 identical test tubes together and place them with an elastic band as shown over here. Put them in an empty beaker. Half fill the 7 test tubes with hot water and put a thermometer into the central test tube and record the temperature for a total of 5 minutes. The student shows this, so this is the results. Prepare and record a table of results as shown in the figure 1.5 as your answer to 1.A1 and your answer to 1.A1. So you need to refer to both this figure and to this first answer. So we know the temperature was 68.5. So let's just write the initial temperature 68.5 degrees Celsius. Alright, so this is 68.5 here, and that's what we need to add. So this is how it should look like approximately. So first test tube was 68.5. Again, units always in the header only. Degrees Celsius, this should be in minutes, that's fine. I would probably say this should instead be time in minutes. So time in minutes. So the second one was 65, then 61, 58, 55. 65, 61, 58, 55. Okay, 67 for the first one, 67, 64, 62, 64, 62, 61 and 60. Then 73, 73, 73. So 3 73s, 73, 72 and 72. Plot a line graph on the grid of temperature against time. Include all 3 sets of data from your table in A11 and a key. Okay, so first draw the axes, number 1 and number 2. Those are your axes. Remember to use the acronym dry mix. Alright, this is essentially something that I used to remember where the independent and dependent variables would go. So D for y-axis, so dependent on the y-axis, independent on your x-axis. Alright, so there's that. So dependent variable, in this case it's a plot of temperature. Okay, never mind, temperature against time. So the dependent variable is the thing that you're measuring, and that is the temperature. Dependent variable, independent variable is the time. That's what's changing or being manipulated. So temperature versus time, and it's a line graph, and essentially there are 3 different sketches that need to be made. So you need to have some sort of a grid looking like this. We have 3 different things, one representative of the first test tube, 3 test tubes, and 7 test tubes. Okay, 1 minute, 2 minutes. So this is dry mix, right? Dependent variable is what we're measuring, and that is the temperature of each test tube. And you put a key on the side like this, where you have, okay, you know, one test tube, don't write it as TT, but you get the point. Test tube, 1 test tube, 3 test tubes, and 7 test tubes. 3, 7. Okay, now what you do is you have Y and X and number 1, 2, 3, 4, 5 for minutes. 3, 4, and 5. 1 test tube is 68.5 in the first minute. So the temperatures seem to be in the 60s, so 50s to 70s range, right? So I would do, and it's also like very specific numbers, so I would probably do something like 20, 40, 60, 80. This is 70, 50, 30. 68.5, 62, 64, 66, 68.5 is about here. Next one was 65 and then 61. So 2, 4, this is, never mind, this is number 2. 65 should be here. And we have 61, number 3, 61 there. Then 58 and 55. 58, 2, 4, 6, 8. So that's there. And 55, which is 2, 4. Sorry about here. Okay. That's the first one. I would probably do it's a line graph. So it's, you know, gonna be a line of best fit. You can make sure the line is really thin and it'll look somewhat like this. Ensure it extends and you try to cover as many points as possible. So something like that. Okay. That's first test tube. Second test tube, let's do a cross. So with 3 test tubes, 67, 64, 62. Okay, 67. First one, 62, 64, 66, 67. I'll probably do a different color just to be safe. 67. Then 64, 62. 64 is here, that's 64. 62, which is there. Then 61 and 60. Okay, 61, which is here, and 60, which is here. And then you draw another line, reduce the width. And yeah, remember it should be a best fit line, so it needs to just pass through as many of the points as possible, and you will know that because there'll be equal number of points on both ends of the line. Okay, so I would put this as a cross like that. And then 7 test tubes. Let's do another color, maybe purple. 73, 73. Well, okay, many 73s, so 73. All right. I'll probably do like a open circle, maybe. 73. 73 for the first one, 73 for the second one, 73 for the third one, and 72 and 72 for the last one. So 72 and 72 for the last one. And then you draw a line, reduce the width. Okay, let's see whether this goes through. Yeah, apologies, it's looking a little bit off. Okay, it should be somewhat like that. Yeah, that seems about right. So this is what your graph should look like. State 2 conclusions for this investigation. So 2 conclusions for this investigation would be, number 1, as time increases or as time progresses, the temperature decreases. That's the first thing you can say. So as time increases, temperature decreases. All right, the second thing you can say is essentially why that's the case, right? State 2 conclusions for this investigation. So why does that occur? That probably occurs because of reduced heat loss, maybe. Like if you look at the fact that temperature decreases, right? Or actually temperature decreases pretty quickly. And what is the setup? Let's look at the setup again. It's actually like an open test tube, so, and there's like no covering. That's the reason why the temperature is decreasing so quickly. So I would say something like essentially the more test tubes, you need to make a conclusion based on the trend in the data. So if I look here, the temperature seems to be decreasing much less when you have more number of test tubes, right? So I would mention that here, the more test tubes, frame it in a way that says, as time increases with greater number of test tubes, the temperature decreases. And this is because having more test tubes allows for reduced heat loss, therefore lower decreases in temperature. State 2 variables that were kept constant during this investigation. You can say the size of the test tube, which is the same, and you can say the let's see what it is again. Have a look at the, always when these types of questions come, always look at the procedure again. So hot water, put a thermometer into one of the test tubes. All of them are placed in a beaker. Yeah, so you can say that actually. You can say that all of the test tubes are placed in a beaker. State 2 possible sources of error in this investigation. So you can say that again, the temperature that that was there at the start, as far as I remember, was not the same, right? Each of the test tubes, if you look at this, the temperature at the start is different. So that could affect how quickly the temperature drops, as seen in the trend line of the graph. So you could say that the initial temperature of the water at the start was not the same. Temperature of the water at the start was not the same. And you can also say, okay, let's see, maybe the surrounding temperature was not the same, but that does seem quite similar to the first answer that you've given. Let's have a look at the procedure again. Okay, 7 test tubes with hot water, 3 test tubes. Yep. You could, okay, because they've not mentioned anything about the volume of water used, you could assume that perhaps the volume of water used was different at the start of the experiment. So volume was not the same in each test tube. Suggest one improvement to the method that was used in this investigation. You could say repeating the experiment. That's one, that I always go for, because this experiment seemed like it was repeated, only once, I think. So you could say repeat the experiment at least twice. Identify, so this just suggests one. Identify one hazard for this investigation. Uses glass test tubes. Hazard is it can break and cause damage. Now, in mammals, respiration releases heat energy to maintain an optimum temperature for enzyme activity in the body. Amylase is an enzyme that catalyzes the breakdown of starch into reducing sugars. Plan an investigation to identify the temperature at which the enzyme amylase is most active. Okay, so this is your design the experiment type of question. For this, you would need to start by identifying your independent and dependent variables. Okay, so in mammals, respiration releases heat energy. Investigation, right? So first thing you can start by doing is listing the independent variable. Now don't write it as IV, write it as independent variable. Independent variable here was temperature. Okay, actually, let's see. Yes, independent variable was temperature. For instance, I would give an example as well. So I would say something like, the case of first test tube, so the case of one test tube, 3 test tubes, and 7 test tubes have at least 1, 2, 3, 4, 5 different temperature measures. So you could say the cases of 1 test tube, 3 test tubes, and 7 test tubes have at least 5 temperature readings in degrees Celsius. dV, dependent variable, is essentially the time taken for the solution, which there were, because remember there was a, which was hot water, okay, heat loss. The actual measurement was in fact your time taken for the solution to turn into a different color, right? Let's see, what was the solution used here? Let's have a look again. Hot water. Believe it was starch, right? So you should reference iodine somewhere. Glass tube. Yep. Oh yeah, here it is. Never mind. So read the question carefully because I almost did not. Amylase is most active. Okay, so it's starch breaking down. And so because of that, you need to reference iodine solution. So the dependent variable is the time taken for the iodine solution to change. All right. Essentially, what you're measuring is the time taken for the solution to change, or the time taken for starch breakdown. Breakdown or disappearance. Yeah, all right. Next one. Variables, you need to mention your controlled variables. Alright, controlled variables, generally I would recommend putting about 2 or 3 controlled variables. So concentration of the starch or concentration of amylase enzyme, concentration of starch, I would probably say here, sorry, concentration of starch, and I would also put let's see, what else can we do? You can say, okay, this seems to be a different situation, right? Amylase in the body reducing starch. In mammals, respiration releases heat. So maybe the quantity of food that was fed to the mammal, the food or plant, of the food plant fed to the mammal. Alright, now you could also talk about other things essentially. So you could talk about, you know, what other things have been controlled. So controlled in the experiment are, for instance, using a colorimeter to measure the color of iodine change, color change of iodine solution. That's one. Always ensure to repeat the experiment multiple times, at least twice is preferable. Repeat your experiment at least twice. And include relevant safety measures. So safety measures could be things like wearing gloves, right? Because you're using iodine solution. Iodine solution is something that can stain, so wearing gloves is a safety measure. Alright. Next, some plants lose their leaves in winter and grow new leaves in spring. Scientists measured a total leaf area of one grapevine plant. They repeated this on 100 grapevine plants and found a mean total leaf area per plant of this. So that's the graph. Suggests why a large number of plants were sampled. You can talk about how this was done in order to get a representative sample or a large enough sample, right? So a large enough sample meaning representative sample. So this is done representative sample, and it just suggests us 1 mark. Estimate the mean total leaf area per plant for month 7 and show how you estimated that value. So month 7, basically here, just draw a line. This is just again an approximate, should be about, you know, 3.3 approximately meter square. Already units are given for you. Using the information from figure 2.1, calculate the percentage increase in the total mean area from month 4 to 6 and give your answer to 2 significant figures. So 4 to 6. So this is 4, this is 6. At 4 you've got 1.4. At 6 you've got 2.2. Never mind. Yep. So the difference is basically 2.2 minus 1.4 divided by 1.4 times 100. This should approximately give you 57%. Let me just check that. .4, yeah, 57% to 2 significant figures. Shows the figure, so figure 2.2 shows a leaf from a grapevine plant. Use the grid to determine the area of the grapevine plant and only count squares that are more than half filled by the leaf and include the unit. Alright, so this was again very similar common question for paper 6, and this is actually quite similar to what I had in my own exam. So how would you know that each one is... Honestly, these questions I think take up a lot of time, I'm not gonna lie, and they're quite like unnecessary. So you could do something like this, just try and highlight what the area is. This should be okay, this should be okay. If you take time to actually count what each is, I would say probably do this. It's about like, make a decent grid like this, right? And then count each separate part. This should give you about 57 is what you should be getting here. So 57 or 59 ish is what you should be getting. Let's just, okay, if you count it correctly, the right number of squares is 59, but they should accept anything in like a decent amount of range, maybe within 2 or 3 squares. And since each square is a centimeter square, that's centimeter square for the unit. So grapevines produce fruits called grapes. A large leaf area is important when growing grapes because leaves supply reducing sugars to the grapes. Describe the method for testing and for reducing sugars. Okay, and include a result for a positive test. So the method for testing for reducing sugars is a Benedict's test. So you just say, mention it as Benedict's reagent test. And it's describe, so you need to mention another thing, which is basically heat or adequate temperature needs to be applied with the reagent itself. A positive result is always color, so the color then generally will be greenish yellow. Okay, this is a photograph from a fig plant. Make a large drawing of the leaf in the figure. So basically there are certain rules for drawing. You need to ensure your outline is very clear, your size is wider than, is or bigger than what the actual image is. You need to have all of the different features. So in this case, it's got 1, 2, 3, 4, 5 of these lobes, and try to draw the veins clearly. I think that is doable, so I am just going to skip this because it's just a drawing. State one similarity and one difference between the grapevine leaf in figure 2.2 and the fig leaf in figure 2.3. Okay, so the difference and similarity. Okay, this one, as you can see, both of them have veins and a stem. Like both of them here have a main stem and then branching veins. That's also that's also the case here, branching veins. That's a similarity. So you can say they have veins, main and branching vein. The difference is that the grape figure, let's see, grape 1, which is this one. The grapevine plant essentially has less veins, actually. Yeah. Yes, actually this one does have fewer veins than that. So you can say the grapevine leaf figure 2.2 has fewer veins. And remember, it's a comparison, it's a difference. So then, or compared to your fig leaf, which is figure 2.3. And that's all for this video, guys. I hope you liked it and found it useful. If you did, make sure to like, share, and subscribe to Biolog, and I will see you guys in my next video. Have a great day.